Hi, hello again. Now we've finished. Well, we've done 7.1, which talks about how the genome is is analyzed, how it's chopped up and sequenced, and so on. Now we've got the sequence. Assuming that we've already got a sequence, the next step is to understand how it works. So the understanding how genome works really is seeing how it is being expressed. And when you say expressed, the subject of analysis will be the RNA, because the genes are expressed into RNA, and then or you can also study proteins, and you'll see towards the subsequent sections you're talking about different approaches that that scientists use to study um, uh, gene expression. Not only do we want to study gene expression, we do also want to study differential gene expression. Part of the approach to study the gene function is to study differential gene expression. And what I mean by differential gene expression is that you want to see the difference between a normal cell and a cancer cells. What are the differences in terms of expression, and that if you compare, and it will pinpoint towards what genes play a significant role in developing cancer. Why do you want to know this? Well, perhaps one day you can use this information to design new drugs, new therapies to control cancer. So expression analysis talks about studying the expression, also comparative analysis, and the approaches used will be described in sequence. All right, so one of the first ones is the source material, the tissue. If you want to see the expression of certain genes in cancer, you must be able to isolate cancerous cells. And the techniques to use this is laser microdissection. Basically, microdissection is cutting very small pieces using laser, right? So, and then having that, and always remember that that this kind of analysis are very sensitive to changes in the environment. So normally, if you do RNA extraction and so on, you want to quickly freeze it in -80, so that you want to retain the information. Otherwise, that your RNAs or might be degraded and things like that. Okay. So study material. This one gives you study material. It gives you high resolution, low resolution. Basically, high resolution, it gives you a greater detail in terms of results, and throughput. That means high throughput means, well, low throughput you're studying one gene at a time. High throughput means you're studying like a whole genome at one go. So these are the differences. And then so it goes on to start to explain the approaches that are used in quantifying. So you're measuring the level of expression. Here it says using of individual genes. When I was doing my research for my PhD research, yes, that's what we were doing. We're studying individual genes. But nowadays, when people do gene analysis, gene expression analysis, they're talking about whole genome efforts. So, but going back to the classical way is to do RT-PCR, reverse transcriptase PCR, or now if you use the real-time PCR, it's called the qPCR, quantitative real-time PCR. But basically, you need to have a good experimental design, good tissue resources, so that you can get the correct cells, and then you start comparing between different cellular states, normal against cancer or treated against non-treated, and things like that. And then you see the different expression. Once you know the different expression of the genes, it allows you to understand the functions, right? Detection methods. If you not well, the newer methods. Previously, when I did my studies, yes, we were using the real reverse transcriptase time PCR, where we did northern blots and so on. But nowadays, most labs will do the quantitative real time, the qPCR, and there are two main approaches to detecting expression. One is to use cybergreen. Cybergreen is a dye that intercalates. It binds to a particular piece of DNA. Now, it only works nice if your, because it is a PCR based, so if your PCR is very specific, that means you've only get one product, right? Your PCRs are very specific, you get only one type of product, and you put cybergreen, the amount of cybergreen that is intercalating will be translated into fluorescence, so the amount of fluorescence can be used to estimate the amount of RNA that's been produced, cDNA. Okay, but the problem with this is that cybergreen binds to any double-stranded molecule, DNA molecule. So that means if your PCRs are not super accurate, your PCRs, if there are contaminations, that means your cybergreen will also bind to this non-intended products as well. So it's not very accurate. So if you really want to study gene expression levels, you are better off using the molecular beacon and TaqMan probes. But basically, TaqMan probes is very commonly used nowadays. What you have is a probe that is designed towards the gene that you want to measure, right? That's the probe. You can see the F and Q. F is the what? Fluorophore, I think, and then Q is the quencher. The Q is, what Q does is Q prevents fluorescence, quencher. So this probe sticks to your target, and when you synthesize, one of the things it will do if you synthesize is that this polymerase will remove the F, and if F, the fluorophore, I think, when it is removed, that means now F is no longer in the vicinity of Q. Q is a quencher, so when it's far, so then this one will be will fluoresce, and you can measure. So the amount of fluorescence will also be the amount of the RNA, and it's very accurate because you design probes that will definitely bind to the sequence that you want to measure. Right? So that's just a bit about qPCR. So I think I've taken too much time explaining qPCR. I'll stop here and then continue with the next video.